So taking strawberry runners is really easy, and you can choose any runner. You can choose big ones with roots already, or you can choose little ones that don't have any roots. This one here has some roots, and this one here doesn't have any roots. It's not even really got leaves yet. So what I do is I get some barbecue sticks and I snap them in thirds, and they're quite easy to snap, and then I build a little bridge out of them by making two sort of 90-degree bends quite close to one another. So now I've got this little bridge here, and that allows me to press the runner down without actually sort of clamping down on the stem. So I just press the runner into the dirt here, grab my little bridge, stick it over, and then press the whole thing down. And you need to press it quite firmly into the dirt so it doesn't move around. Roots down, leaves up every time. Now if you've got a little runner coming off the runner, we're just going to get some scissors here and just chop that off, because we want all the energy to stay with the runner rooting. It doesn't really get any easier than that, does it? Just remember, leaves up, roots down. If it doesn't have roots, just make sure the leaves are pointing up. Make sure they're not trapped under the skewer. Keep the soil nice and moist. It's good to keep them attached to the mother plant until the roots actually appear, otherwise they'll just wither and die. And if you want to have strawberries from the runners this year, if you catch them early enough in the year, so sort of before July, keep the umbilical attached, and you may get some strawberries from these runners before the end of the year. If not, then you're just going to need to cut the umbilical once.
I'm just a heap of human wreckage. I'm just a human toxic freak. I'm schizophrenic, no I'm not. I'm schizophrenic, no I'm not.
تكاثر الهيدرا بصورة سريعة لا جنسيا عن طريق التبرعم. هذا الهيدرا وبعد يومين من بدء التغذية كون له برعمين، وخلال 24 ساعة كونت البراعم لها زوائد أو مجسات، وبعد 48 ساعة انفصلت تلك البراعم لتصبح هيدرا. ولكن ما هي خطوات حدوث التبرعم؟ يبدأ التبرعم بتجمع الغذاء في منطقة في أسفل جسم الهيدرا فتكون انبعاج، يلي ذلك انقسام لخلايا الخارجية الإبيديرمس والداخلية الجاستروديرمس، ويلي ذلك تكوين تجويف لهذا الانبعاج الذي يكون له مجسات ثم فم، فيبدأ بالتغذية. بعد ذلك يحدث تخصر في نهاية هذا البرعم ثم ينفصل عنه، وقد يقوم البرعم الجديد بالمساعدة في الانفصال بسحب نفسه من الخلية.
Generate lost limbs. Here is a starfish from the genus Phoromia. We took him in as a rescue, having lost all but one arm. Since then, this little guy regrew 5 arms and is starting to look kind of like a little hand. Starfish do not have a centralized brain, but they do have very complex nervous systems. They are able to sense what they touch with their feet, as well as detect light from the tips of their arms. Because they have in essence a distributed neural network, starfish cannot plan actions in advance. They respond to stimuli, and the arm that is first to detect something becomes dominant and overrides.
In prophase 1, the DNA coils tightly and individual chromosomes become visible under the light microscope. Homologous chromosomes become closely associated in synapses, and they exchange segments by crossing over. By metaphase 1, the nuclear membrane has disappeared, and the microtubules form a spindle. Spindle fibers attach to only one side of each centromere, and the two homologous chromosomes attach to microtubules orienting from opposite poles. Each pair of homologs then lines up on the metaphase plate. Either maternal or paternal homolog may orient toward a given pole. In anaphase 1, the microtubules of the spindle fiber shorten and pull the chromosomes toward the poles, taking both sister chromatids with them. Each pole ends up with a complete haploid set of chromosomes consisting of one member of the homologous pair. During telophase 1, the nuclear membrane reforms around the daughter nuclei. Each daughter nucleus contains two sister chromatids for each chromosome attached to a common centromere. Because of crossing over, the two sister chromatids are not identical. During prophase 2, the nuclear envelope breaks down and a new spindle forms. In metaphase 2, spindle fibers bind to both sides of the centromeres. During anaphase 2, the spindle fibers contract and the sister chromatids move toward opposite poles. In telophase 2, nuclear envelopes reform around the sets of daughter chromosomes.